Good afternoon, everybody. Today I want to talk about microscopic analysis of blood. As you know, the average of blood in the adult is about 5 liters. And you know that the blood is the liquid connective tissue. All types of connective tissue is composed of cells and extracellular material. In the blood, the cells are called formed elements. We call them red blood cell or erythrocyte, white blood cells or leukocyte, and platelets or thrombocyte. And the extracellular material or matrix of the blood has only ground substance. The ground substance in our blood is liquid, and we call it plasma. In the plasma, we have different dissolved protein, respiratory gases, regulatory substance, nutrient, waste product, and electrolyte, and many more substances in the plasma. We don't have any fibers when our blood is in the healthy blood vessels. When the blood vessels become sick, or when we have tearing in the blood vessels, when we have any cut, at that time, the fibers are created in our blood from the fibrinogen protein. And after that, we have the process of blood clotting, which you will learn about it in the part of hemostasis or blood clotting. Today, I want to talk about the blood cells for you. When we want to check the blood cells, we need to make and prepare blood smear. For making blood smear, we can poke the finger and we can collect one droplet of blood, put it on the glass slide, and with the other glass slide, we can spread the blood on the surface of the first one. Then we can have a very thin film of blood on our slide. We should put it to dry, and after that, we can stain it with different staining procedures. Then we can check the blood cells. We have different types of blood cells, which I talked about it before for you. You can see erythrocyte, platelets, and leukocyte. Our leukocytes are very different. We have two groups. One group is called granulocyte, and one group is called agranulocyte. Granulocytes are divided into neutrophil, eosinophil, and basophil, and agranulocytes are divided to lymphocyte and monocyte. We have different types of pathogens which invade our body. So we need different types of white blood cells to fight with these pathogens. In the blood smear, you can see the different shapes for the blood cells. You can see red blood cells. You can see small fragments which are platelets. And also you can see different white blood cells like monocyte, eosinophil, neutrophil, lymphocyte, and basophil. The first cell which I want to talk about it is the erythrocyte. The erythrocyte is a flat cell which contains biconcave surface. This is the surface view, and this is the side view or sectional view of red blood cell. In the human, the red blood cell has 7.5 micrometer diameter, and the thickness of our red blood cell is about 2 micrometer. But this thickness is not the same in all parts of red blood cell. The center of red blood cell is much more thinner than the peripheral part. So the accumulation of hemoglobin in the center is less than peripheral. So we can see the center of red blood cell paler than peripheral. This is the normal shape of red blood cell. The red blood cell doesn't have any nucleus, so it cannot do any mitotic division and any protein synthesis. It doesn't have mitochondria, so it can make ATP anaerobically by glycolysis. And most of the organelles are lost during development. Here you can see the side of red blood cells. You see that we have biconcave disc. The center is much more thinner than the peripheral part, and this shape can help the red blood cell to bend, to fold, and pass through the tiny blood vessels, and also it gives the red blood cell a greater surface so it can attach to more oxygen. The function of red blood cell is carry oxygen from lung to all parts of the body. And also the red blood cells can pick up about 23% of carbon dioxide from different organs and bring them back to the lung. The carbon dioxide attached to globin part of hemoglobin, while the oxygen can attach to iron ring. Here you can see the normal shape of red blood cell. As you see here, the center is thinner than the peripheral, and the border of our red blood cell is completely smooth. It's important to pay attention to the shape of red blood cell. If the red blood cell become bigger than normal, we have a problem which we call it macrocytosis. It become smaller than normal, we call it microcytosis. If the size of red blood cells is different in our blood smear, we call it anisocytosis, and again, it's a problem. If the color of red blood cell in all parts is homogeneous, this is the other problem, which we call it spherocytosis. So you see that paying attention to the shape of red blood cell can help the technician in the blood test lab to identify different diseases, different problems in the patient. Here you can see a blood smear. In this blood smear, you see that some of the red blood cells are normal like this, but some of the red blood cells have homogeneous color in all parts. So this red blood cell is not normal. Some of the red blood cells are crescent shape, like this one, or this. This smear is from a patient who suffers from sickle-cell anemia. When the body wants to make hemoglobin, we have a mutation in making hemoglobin. This is a genetic problem. Instead of glutamic acid in beta chain, we have valine, and the valine can change the shape of hemoglobin. When the three-dimensional shape of hemoglobin change, you can see the red blood cell in this picture, sickle cell anemia. So paying attention to the shape of red blood cells in the smear is very important. The other problem which we may have for red blood cells are crenated red blood cell. Normally, the border of red blood cell should be smooth. If you have crenation in the border of red blood cell, it means that we have infection in our body. So crenated red blood cell is not normal. Now I want to talk about different types of leukocytes or white blood cells. I told you that the white blood cells are divided into two categories, granulocytes and agranulocytes. The granulocytes are the white blood cells who contain granules. But what is granule? Granules are the lysosomes which we have in the cell. And you know that the lysosomes contain digestive enzyme. Each of this group of white blood cell contain different types of enzyme in their lysosomes. So they have different colors, different names, and different functions. We call them granulocyte, and all of them have phagocytic activity. The other name of this group of white blood cells is polymorphonuclear cells. The meaning of polymorphonuclear means they have different parts or different lobes for their nucleus. And now I want to talk about it. The first cell which I want to introduce to you is called neutrophil. The neutrophil means it has granules which are neutral. Look at here. This is the cell membrane of neutrophil. Inside the neutrophil, you can see a nucleus which contains different lobes. The number of the lobes of the nucleus is between 3 to 5. It's important. And as I told you, the color of granules in this cell is neutral. Neutral means it doesn't have affinity to red color or blue color. So the granules or the dots or the lysosomes which you can see in this cell are pale purple or lilac pale. The next cell is called eosinophil. The eosinophil is a cell which contains bilobed nucleus. It means that the nucleus contains 2 lobes, but the granules in the cell are a scarlet color or rosy orange color. So you can see red granules in the eosinophil. The eosinophil is important for fighting with parasitic infection and also allergic reaction. Pay attention to the number of the lobes of the nucleus. They contain only 2 lobes. And the last cell which we have in this group is called basophil. The basophil has the S-shaped nucleus. If I want to draw this nucleus, it's like this. The nucleus is S-shaped, and in the cytoplasm, we can see the granules which are dark blue. The dark blue granules sometimes cover the nucleus, and you cannot see the shape of the nucleus clearly. The basophils can make inflammation in the body. When we have cut, when we have trauma, when we have injury in any parts of our body, the basophils migrate to that part and they can release histamine and heparin from their granules and they can make inflammation. In the immune system, I will talk about it for you, why the inflammation has benefits for our body, why the inflammation is one function of immune system in our body. So here you can see the S-shaped nucleus and the dark granules which we have in the cytoplasm. So the first cell in this picture is neutrophil, then you can see eosinophil, and the last one is called basophil. All of these cells are called polymorphonuclear cells because the shape of their nucleus has different lobes. The next group of white blood cells are called agranulocytes. The other name for agranulocyte is monomorphonuclear cell. Means that they have only one part for their nucleus, mono part or one part. In this group, we have lymphocyte and monocyte. First, I want to talk about lymphocyte for you. The shape of the lymphocyte is like this. The lymphocyte first is very small cell. Then it contains a large dark blue nucleus, as you see in this picture. This is the nucleus. Actually, the nucleus occupies all parts of the cell, as you see here, and you can only see a very thin rim of cytoplasm around the nucleus. So the purple part is cytoplasm, and the blue part is the nucleus of your cell. So you see that the ratio of nucleus is much more than the cytoplasm. The cytoplasm is very thin around the nucleus. We have two types of lymphocyte, B lymphocyte and T lymphocyte, which I will talk about it in lymphatic and immune system for you. The lymphocytes can fight with viruses. The next cell which we have as the agranulocyte is monocyte. The monocyte is the largest white blood cell. This cell has kidney-shaped nucleus or bean-shaped nucleus. The monocyte cannot live in the bloodstream for a long time. When the monocyte is created in the bone marrow, it can come to the bloodstream, live in the bloodstream for 3 to 6 hours, and then go out of the bloodstream and go to the connective tissue. When the monocytes go to the connective tissue, we call them macrophage. And you know that the macrophages can do phagocytosis. We have different names for the macrophages in different parts of the body. In the bone, we call it osteoclast. In the brain, we call it microglia. In the skin, we call it Langerhans. In the liver, we call it Kupfer. In the lung, we call it dust cell. But all of them have the same function. All of them can do phagocytosis. So pay attention, in the group of agranulocyte, we don't have any visible granules in the cell. They have monomorphonuclear, just they have one piece for their nucleus. In the lymphocyte, the nucleus is round, while in the monocyte, the nucleus is kidney shaped. Here in this picture, you can see neutrophils. As you see here, the color of granules in the cell is lilac or very pale purple. They contain between 3 to 5 pieces for their nucleus. In this one, you can see 1, 2, 3, 4. And in this one, again, you can see 4 pieces of the nucleus. The next one is called eosinophil. The eosinophil has bilobed nucleus, and the color of granules in the cell is scarlet or red. And the last one is called basophil. The basophil has dark granules, and these dark granules can cover the nucleus, and we cannot see the shape of the nucleus very clear. The basophil can make histamine and heparin, which can make inflammation for us. And in this picture, you can see a granulocyte. We have lymphocyte and monocyte. The lymphocytes have large nucleus and thin rim of cytoplasm around the nucleus, while the monocyte has kidney-shaped nucleus and larger amount of cytoplasm. The lymphocyte can fight with viruses, and also they increase at the time of different types of tumors and cancers, and the monocyte can fight with chronic infections. And the last cell which I want to introduce to you is platelets. The platelets are not actually a cell. A platelet come from a large cell in our bone marrow, which we call it megakaryocyte. The megakaryocyte is a large cell in the bone marrow. The cytoplasm of megakaryocyte breaks down to small pieces. Each of these small pieces is one platelet. The platelets are very smaller than the red blood cells, as you see in this picture. The platelets doesn't contain any nucleus. This is a model which we have in the lab for the shape of the blood cells. In this picture, you can see number 1, they are red blood cells. Number 2 represent lymphocyte with large nucleus without any granule. Number 3 is the monocyte, because you can see horseshoe nucleus or kidney-shaped nucleus, and this is the largest white blood cell. Number 4, both of them are neutrophil, because in the first one you have 3, and in the second one you have 5 pieces for the nucleus, and you don't have red or blue granules in the cell. Number 5 is eosinophil. You have bilobed nucleus and red granules in the cell. And number 6 is basophil. You have dark granules which cover your nucleus. And number 7 are small fragments which represent platelets for us. We have a mnemonics which is very helpful when you want to talk about white blood cells based on their numbers. Never let monkeys eat bananas. N is neutrophil. L is lymphocyte. M is monocyte. E is eosinophil. And B is basophil. These are the order of leukocytes based on their number. The neutrophil is the most numerous, and the basophil is the least white blood cells in our bloodstream. We have a formula which we call it differential white blood cell count. But why do we need to have differential white blood cell count? When you go to do blood tests, the technician makes the smear of your blood, and then in the smear, she or he starts to count the number of your white blood cells. For example, in your blood smear, they count the neutrophil, they are 122, the number of lymphocytes is 58, monocyte is 14, then eosinophil is 6, and they cannot find any basophil in your blood test. Then they want to make conclusion about your test. They want to see if you have any disease, any problems, or you are completely healthy. So they need to do differential white blood cell count. For doing this, we have a formula: the number of each white blood cell over the total number of white blood cells times 100. This table, for example, is from one patient. And now I want to calculate the diff count for neutrophil. If I want to do so, you can see that the number of neutrophil is 122. The total number of white blood cell means you add up all of these numbers together, add all of them together. It becomes 200. And then you multiply this fraction with 100. When you do this equation, the result is 61%. So this is the percentage of your neutrophil. For the lymphocyte, if we want to do it, we have 58 lymphocytes in this blood smear. The total white blood cells are 200 and multiply by 100. The result is 29%.
Excuse me, Professor, where did you find the 200?
If I want to calculate the diff count for monocyte, I have 14 over 200 times 100. So the diff count for monocyte is equal to 7%. And if I want to do it for eosinophil, it is 6 over 200 times 100. And it becomes 3%. And as you see here, we don't have any basophil, so the result is 0% for the basophil. So when you want to have differential white blood cell count, you need to have the number of white blood cells, each of these white blood cells, over the total number of white blood cells times 100. And this percentage can show you the percentage of each of them. Now, how can we use these percentage? Okay, when we did the previous table, the diff count for neutrophil was 61%. For lymphocyte, it's 29%. For monocyte, it's 7%. Eosinophil is 3%, and basophil is 0%. Then we need to check them with the normal value. You have the normal value of all white blood cells on page 19 in your lab manual in the second table. The normal value for neutrophil is 60 to 75, and our result is 61. So the neutrophil of this patient is normal. If the neutrophil is above 75%, it means that that patient has bacterial infection, for example. The second one is lymphocyte. You see that the normal value of lymphocyte is 20 to 30%. And here the lymphocyte is 29%. So again, it's normal. If the lymphocyte is more than normal, it shows us some problem, like viral infection, like heart attack, sometimes during pregnancy, sometimes during different cancers and tumors, we have more number of lymphocytes. The amount of monocytes, the normal value should be 3 to 8%. And here we have 7%, so it's normal. If the monocyte is more than 8%, it means that we have chronic infection. The eosinophil is 1 to 3%, and ours is 3%. So again, we are in the normal value. If it's more than normal, we have parasitic infection. And the normal value for the basophil is 0 to 1%. And we have 0%. So again, it's normal. So all the times when you do blood test, the technician counts your white blood cells, and then they make the percentage of them and check them with the normal value, and then send the result to your physician. And based on the normal value, the physician can identify you are normal or you have any bacterial, viral, fungal, parasitic infection, any cancers, or any other problems. This is how this differential count of white blood cell works for us. The first slide, you see that the red blood cells are completely normal because they have a smooth border, and the center of red blood cell is paler than peripheral part. So the shape of our red blood cell is normal. Here you see that some of the red blood cells overlap on each other, so their color become a little darker. It's due to overlapping. It's not a problem of red blood cell. Again, you can see the normal red blood cell, a smooth border, central pale part, and peripheral darker. In this picture, we have problems for our red blood cells. You see that some of the red blood cells are crescent shape. Some of the red blood cells doesn't have central pale part, and some of the red blood cells has crenation. So this smear is from a patient with sickle cell anemia. You see that we have different shapes for red blood cells in this picture. In this picture, you have red blood cells. Some of them are not normal, because you can see crenation in the wall of red blood cell. This one, and this one shows crenation, and also this. Between these red blood cells, you can see platelets. These small fragments are called platelets. Here you can see the formation of platelets, which I showed you before. We have a large cell, which we call it megakaryocyte in our bone marrow. The megakaryocytes can make some feed process, and then these feed process breaks down to small fragments. Each of these small fragments is one platelet. So the platelet is not actually a cell. It's a piece of cell without any nucleus. In this picture again, you can see red blood cells with central pale part with a smooth and soft border and the platelets between them. Here you can see normal red blood cells. Here they overlap on each other. When they overlap on each other, we call it rouleau formation. They make the structure like Rolex necklace. They are overlapping on each other. And here you can see neutrophil. We call the neutrophil polymorphonuclear cell because it has more than one part for the nucleus. Here we have 1, 2, 3, 4, and 5. And only our neutrophils have 3 to 5 parts for the nucleus. In this picture, you can see red blood cells. Your red blood cells are not normal because they don't have central pale part. Just this one is normal and this one, which has central pale. And these two cells are neutrophils. As you see here, you can see 1, 2, 3, 4. And in this one, you have 1, 2, 3, 4, 5 pieces for the nucleus. And the color of granules are very light purple. Again, you can see red blood cells and neutrophil, 1, 2, 3, and 4 pieces for the nucleus, and light purple granules in the cytoplasm. This is neutrophil based on the information which I gave you before, and this is the eosinophil. You have 2 parts of the nucleus and the red granules inside the cell. This is eosinophil. You have 2 pieces of nucleus and red granules. These are red blood cells, and this is platelet. Again, here we have eosinophil, platelet, and red blood cell. Eosinophil and red blood cell, and these 2 are platelets. Again, you can see eosinophil here, red blood cell, and platelet. This is basophil. You have large, dark granules which cover your nucleus, and you cannot see the shape of nucleus. Again, basophil, dark granules. Again, you have basophil with dark granule. Basophil. And this one has only 1 round nucleus. This is lymphocyte. It doesn't have granules, and you can see very thin cytoplasm around the nucleus. Lymphocyte with 1 round nucleus and cytoplasm around. These are red blood cells, and this is platelet. Again, lymphocyte. And this is monocyte, the kidney-shaped nucleus. Here you have 1 monocyte, and this is neutrophil. Here you can see platelet, and these are red blood cells. These are the blood smears and the blood cells. I will talk about blood test next time for you.